Right, well, good day, folks. It's good to see you all, and thank you very much for all of those who are in the chat already. Good morning, good evening, good afternoon, wherever you're joining us from around the world, and welcome back to Creation Conversations, where we have a slightly more limited team tonight. John is unable to be with us because he is over at Jurassic Ark, doing some great work with our curator over there, working on some new research projects. And Sam is unable to join us as well tonight, so I shall be looking after behind the scenes. So yes, thank you for your donations that have already come through. Thank you for your questions. We will certainly be able to get to some extra questions tonight as we're a little bit slimmed down. And up until about 5 minutes ago, we had no Glenn, and we thought it was just going to be the three of us. But Glenn did manage to get in. There's been no electricity where he is, and the electricity suddenly came back on. So do all pray that Glenn's electricity stays on for the next little while, because tonight's topic is this sort of designer genes. Are we designed? Are our genes designed? How is our DNA designed to evolve or not to evolve? That's what we're really going into, and we've got examples from castles and examples from all sorts. So it's going to be great fun tonight. But because Glenn is sort of on questionable Wi-Fi connection, we're going to start with him and dive straight into things with Glenn's program and presentation as we move forward. And then we've got stuff from Craig as well as from Diane. So it's going to be a great night. And yes, do keep getting in your questions and we will deal with them after a little bit of time. We'll have a good couple of times for questions. So Glenn, if you're all ready, it'll be straight over to you if you want to pull your slides up to give us our first session of this evening.
Well, as Joe said, it's been a little interesting to just getting online. I was all driving to a supporter in our ministry just so I could join y'all, and got to their house and they had no electricity. So I rushed back and called Joe and through my daughter's phone, and then electricity came back on. So here I am. So we're going to start off with just reminding everyone, what does evolution teach? It teaches, that you start with this pool of chemicals and things just somehow seem to come together magically. And from that, you start getting this progression of more and more advanced life forms. But what does God's word say? In the very first chapter, it says 10 times that things are made to reproduce after their kind, after their kind, after their kind. God makes it very clear. He gets the point across. And so we're going to talk a little bit about that and about the DNA involved in that. And my question to the audience is, can natural processes by random chance code for this descent, which is actually an ascent from a common ancestor to more and more advanced life forms? You're going to hear a lot more about this from others about DNA and RNA, but what we know about it is that it's got these phosphate groups and these sugars that are what forms the codes, and that they link the DNA double helix together. And that is the codes, the instructions for the body to develop and function. And the RNA serves to transfer and translate and copy this DNA. I'm going to talk about the coding because when I was in college, that was a long time ago. Let's see. In 78, I took Fortran programming because you had to have a language requirement. Well, they allowed, instead of me taking Italian or French or, something else, German, I took Fortran and learned computer programming. And that was just posing the instructions to tell the computer what problems I wanted it to solve. I was doing my research on soil physics, and so it was involved computer programming. Well, the DNA is sort of like a computer program that tells the computer what to do. And then the RNA has to transcribe that sequence of instructions into machine language. And That's zeros and ones. The code is zeros and ones instead of the DNA, which is A, C, G's, and T's, and the RNA is A, G, C's, and U's. Okay. So this was so long ago. You guys, I'm hoping the audience will get a kick out of this. We didn't have personal computers. There's no such thing. I had to go over to the computer science department and I would write my code out and I would have to type it into a machine that punches cards. And each card was a single line of Fortran code. And so to have a program, which would usually involve a few hundred codes, that's a few hundred cards. Okay. So these cards then are like the sequence in the DNA. But then I had to run the cards in the stack of cards through a card reader, which would transfer that into the zeros and ones in the computer for the computer to be able to process what I was wanting to tell it to do. Well, that was quite involved and quite aggravating too, having to walk across campus. I remember my sister was one of the higher ups in the computer science department, and my major professor used her to get a meeting with the computer science department to ask them if we could get a computer in our building. And their response was, what would someone in agriculture and soil science need a computer for? That was the kind of the state of, now we can't imagine doing life without a computer. So what did I learn? Well, I learned don't drop your stack of cards because you're not going to get any good code. Other lessons learned. Just a single little punch and a single card and your program is going to fail. If you just made one tiny mistake, the program fails. Even with time, those punch cards and the little punches will get damaged. And as with time, those damages will end up making your program fail. And if you make a mistake in the writing of your code, you get off just a little bit, you can produce results, but those results will be just meaningless results, much like mutations. Well, keep in mind, NASA scientists and engineers have sent people to the moon with computer programs using this binary code of just zeros and one. But DNA and RNA is a base 4. It's a quaternary system. It is so much more complex. And that's what I wanted to kind of give you a feel for. If you take just the 0 and 1s and look at the combinations, you can combine these into 0, 0, 0, 1, 1, 0, 1, 1. It's 8 letters, 4 combinations. But if you're working with the base 4, such as DNA, this is your combinations of this. The simplest combinations would be 264 letters and 80 different combinations. The complexity of the DNA is just mind-boggling. And then when you get just the simplest sentence using those combinations, I've given you just an example. If that's not complicated enough, you think of the ones and zeros are read in one direction. The computer only reads those simple codes in one direction. DNA and RNA, they're read frontwards and backwards. It's mind-blowing, the complexity in the DNA code. And then to think that all of this, the entire human genome composed of 3 billion letters, can fit inside a microorganism. And when you think of how tiny a microorganism is, when you look at a dot on a page, that's bigger than the microorganisms. It's called a microorganism for a reason. It's a microbe. So I love this quote. This comes from a book by John Morris, who has passed away now, but was head of Institute of Creation Research. He wrote a book, Is the Big Bang Biblical? 99 Other Questions. And in that, he had an interview. He got the transcripts from an interview from a person writing a newspaper who did an interview with a molecular biologist who studied DNA codes and tried to decipher DNA codes. And he asked the person, do you believe that the information evolved And the biologist said, George, nobody I know in my profession believes it evolved. It was engineered by genius beyond genius, and such information could not have been written any other way. The paper and ink did not write the book. Knowing what we know, it's ridiculous to think otherwise. And so I was going to end with that. It's clear these natural processes, by just random chance, cannot code for life. It took a genius of genius. It took God. A binary system is so much easier than a quaternary system. So with that, I am going to end. And Joe, we can go back to you.
That's great. Thank you for that there, Glenn. That's wonderful. And so basically what Glenn has covered then is this concept that information, computer coding, has to come from what is effectively an intelligent source. You can't get codes and the like, whether it's a computer code or whatever, from anything other than an intelligent source. But what about the concept then of things like DNA and so on and so forth, and moving into the more sort of biological side of things? Well, to make that link from what Glenn's been speaking about into what Diane will speak about, which is the more sort of biological genetic side of things, Craig, you've got a nice sort of practical example for us.
Yeah, well, I'm not exactly sure I totally agree with Glenn there, because I've got a great toy I want to show you. It's a castle set that I grew up with, and I really loved it. It's got a whole bunch of specific blocks in it. So there's like a castle tower piece. It's got some rounded pieces. You can make rounded towers. It's got little windows that you can build things out of. Top of a tower, the doorway, and all these sorts of specific building blocks, a round tower. And Glenn was sort of saying that random processes don't do anything with codes and design and so on, but all I have to do with this is shake it a bit, random processes, a little bit of time, a little bit of energy, a little bit of time, that's it. And just a bit of patience, and then oh look, it's already made this fantastic castle for me. Wow. It's amazing what a bit of random process can actually do. So this was a lot of fun. There it is. The windows are all in place. The towers are all in place. That's just been a great toy, and it builds all sorts of different castles for you. And it's just random process that does it. Now, of course, that's not true. And even a 4-year-old watching this would realise that that's not going to happen on its own. Even though there's specific blocks that are meant to come together to do that, it's never going to do it on its own. So really, the difference between, obviously, I had that one and I had that one, and the only difference between those two is information. It's intelligence, because you've got to get those blocks, those building blocks, which are designed to come together. You've got to get them together at the right time and in the right order. The foundational ones first, build it up around that, think about it, how you're going to do it, and you'll end up with something that looks like a castle, hopefully. Now, you can use those very same blocks to build a whole bunch of different castles, but every single one of those castles needs a creator, needs the intelligence. So when we see similar sorts of proteins from similar sorts of genes throughout creation, that doesn't mean that they're linked by random processes. It just means the creator has used similar proteins for similar sorts of purposes throughout his creation. Now, the strange thing is that many people think that non-living chemicals, proteins, we know them as proteins, will come together and do life on their own. And I think this is crazy. We know that that doesn't do things like those castle blocks, remembering that they are meant to come together. They are designed, those blocks. One block is really a design because plastic doesn't do blocks on its own either. But those blocks are designed to come together, and yet they won't without a mind organising them. So that was a toy that I loved playing with over many years, and I built many castles. Maybe it's my English side coming back out of me, not just the colonial that's living down here in Australia.
There you go. All right. So we've gone from two very physical examples, right? We've gone from Glenn and his computer coding to Craig and his castle building. But let's try and shift this then over into a more sort of a biological aspect. We've got the concept. Right, things don't happen by themselves. You don't get codes, you don't get structures, you don't get clearly designed things purely by random chance. So let's ask this question then. If we go into the question of genes, right, and our topic today, designer genes designed, in fact, to not evolve. Well, Diane, you're our resident sort of biological and medical expert. So you're going to take this next little session, and then we'll have a break for some questions. We've already got some questions coming in. So thank you very much for that. Do keep those questions coming. That's great. And we'll deal with them after a little bit. But Diane, over to you now to talk about this question of designer genes and to try and take the points that Glenn and Craig have made about computer codes and castle building and fit them together into a question of biological design. So over to you.
Yes, indeed. Yes. Well, those two illustrations we've just had are very good. And the punch cards, and I'm actually old enough to just remember punch cards, right? They don't write the code. They carry the code.
Yes.
The same with the castle. The design of that comes from outside the castle. The plastic components don't actually make the information needed to put it together, even though it does go together. So perhaps if we could go to my slides now.
They're all up and ready for you.
They're all up and ready. That's good. Yes. So when we look at genes are information, but the information has to be carried on some sort of substance. And it is very clearly designed not to evolve when you look at how it works. And the interesting thing is the secular scientists do actually recognize that life is made out of information. And every now and again, they give themselves away. This was from an article in the Proceedings of the National Academy of Sciences. All known life on Earth is based on biological polymers. Now, that just means long strings of long molecular strings. So long strings of small molecules stuck together to make long molecules, right? And these act as information carriers and catalysts. That's molecules that help other chemical processes carry on in the right place at the right time. Therefore, any theory for the origin of life must address the emergence of such a system. In other words, where does the information come from? Now, an Australian man named Paul Davies put this in a little bit more blunt manner. Being an Australian, we tend to be rather blunt. And he asked this question: how did stupid atoms spontaneously write their own software? And where did the very peculiar form of information needed to get the first cell up and running come from? Because no one has ever seen non-living chemicals spontaneously turn into a cell. The genetic information that we study is information that is already in a cell. And if you look at a cell, it has a store of information inside it called the nucleus. And inside of that, there is about 2 meters of DNA, right? That's an awful lot of chemical information, all very, very neatly bundled up and packaged and kept inside that cell. So how is the information actually stored? Well, it's stored as small chemicals, right? And these small chemicals are like the letters in the code. And we can compare it with any sort of language we like. Now, the chemical letters are A, T, G, and C. Now, these are some small molecules named adenine, guanine, cytosine, and thymine. You don't need to know that. We'll just stick to the A's, T's, C's, and G's, and they are just small chemicals. And like the letters in a word, right? The individual letters are quite small. It's not until you put them together that you get any meaningful information. Now, they need to be held together in a structure, and that's how we get the very famous double helix that everyone knows about. And there's a very interesting way that they fit together. They fit together by a process that's called base pairing. Now, that the individual letters, that those molecules are called bases. So a DNA molecule consists of two sort of long strands of molecules called deoxyribose nucleic acid, which is a sugar and a phosphate, and that forms the backbone of it, the two strands. And then the bases project into the center between the two long strands, and they form a weak chemical bond, but it's enough to hold it all together. But it can be broken quite easily, and that's important when it comes to actually copying it. Now, there's a very interesting way that they fit together. The A's and the T's all fit together, and the C's and the G's. So if we have our four letters like this along one strand, right, A will always pair with T. So if you have an A on one strand, the opposite strand will have a T. If you have a T, it will always pair up with an A on the other strand. And the same goes for the C's and the G's. They will all link up together. And the reason for that is that is how the information can be copied accurately. Now, every time a cell divides, the information in your DNA has to be copied, and it must be copied accurately. And because of this base pairing, that's how that can be done. And the way this works is that if you have your two parallel strings of information with the base pairing, right, A's and T's, C's and G's, right, you can then split apart the two strands and make a new copy. And so we now have two identical copies using one half or one strand as the template for the other. Now that happens every time one of your cells divides, and that's going on sort of millions and millions of times all the time in your body. And it's essential that that information is copied accurately, because otherwise your cells won't work. So let's go back to the cell. Well, that's the information that's stored in the nucleus, but in order to be used, it has to be somehow transported. The information has to be transported out into the machinery, into the working part of the cell. Now, DNA stays in the nucleus. It's important that it is kept there. It's like an archive of information, and it needs to be protected. But in order to use it, the information needs to be sent out into the cell. Now, it's a bit like if you imagine a building site, you don't take the architect's original drawings out to the building site, right? You take copies. And this is what happens inside a cell. Now, this is a very simple way this happens. So DNA is an archive of information that is packaged up and stored. If you need that information, you make a working copy. And that is the molecule that Glenn referred to, RNA, and it is a working copy. So it's the same information, but it's not exactly the same substance. In fact, it's only a single strand, and that information then has to be sent to a protein-making machine out in the cell, which is called a ribosome. So the RNA is sent to the ribosome, which grabs hold of it, and it will literally read the information, that series of chemical letters, and use that to make a protein. Now, proteins are the the stuff of life. Actually, they form the structure and the function of the body. So this is how we go from DNA information to a physical cell and a physical body, is basically by this process. Now then, there are millions of other chemical processes that then happen around that, but we'll just stick to this first. So what happens with the RNA information? It is a similar molecule to DNA, but not exactly the same. Now, in fact, it is made by base pairing as well. That's how the information gets transferred, right? A bit like copying the DNA, but instead of having T's, right, chemical letters called T's, it has a chemical letter called uracil, so we'll call that U, and A is paired with U. So in order to transfer information from DNA to RNA, we have a similar process, and we end up with that same information, but just slightly different letters. Now, we have transferred that information to different chemicals, but it's still the same information. And that's important to understand the difference between information and this substance that actually carries it, because the substance didn't make the information, it is carried on it. So we now have information being transferred. Each step involves different molecules, but the information is the same. It doesn't come from the chemical properties of the molecules. It's actually in the sequence of molecules, just like the letters in a language. In order to make a word, right, you can have 26 letters, but they can be put into all sorts of different combinations to make hundreds of thousands of words. I'm not quite sure how many words the English language has, but I think it's in the hundreds of thousands, certainly in the tens of thousands. So we are talking about something that behaves like a code. So hence the analogy that Craig and Glenn have used. We are talking about a code. We're talking about information where the components that carry it didn't generate it. They just carry it. But we have an interesting problem with DNA code and RNA code, right? Body structure and function requires proteins, which are then used to make all sorts of other molecules, as well as being the body structure, right? And proteins are made up of small molecules called amino acids. So a lot of biological molecules are small molecules strung together to make bigger ones, just like the components in Craig's castle. You've got a whole lot of small components that can be assembled in different configurations to make a different kind of castle. Now, there are 20 different amino acids, so you can get all sorts of amazingly varied shapes and sizes of proteins. And the information that's carried on DNA and RNA actually codes 4 amino acids. But the interesting thing is we've just said that there's only 4 DNA letters or 4 RNA letters, but somehow we've got a code for 20 different amino acids. So how is that done? Well, there is a solution, and to help us understand that, let's look at code that's carried in English language, in plain language. All right. Now, what does that say? What does that say? Any of you know?
The cat ran off the mat.
Right. Yeah. Yes, you're right. Now, how did you work that out?
I suppose I already knew the letters, the words within that.
That's right. Yes. You recognized combinations of letters. It's not just a random lot of letters. You looked at that and you said, yes, there are particular combinations that go together. And what you've done is you've separated out the combinations, the 3-letter combinations, using spaces, right? And we go from what could be just a random collection of letters that's been thrown together into a meaningful sentence. Now, what you've done there is apply what's called a reading frame, right? We could keep all of those letters together and just split them up with lines, right? In written language, we use spaces as a reading frame. Now, in DNA and RNA letters, right? They're all stuck together. They don't have spaces together. But in order to make that information meaningful, it does have to be read in groups. Now, they happen to be groups of 3 in English. Of course, we can have groups of 2, 3, 4, 17, however many you like. Now, here's an interesting problem. What happens if you change the reading frame? Right. So if we were to just move it a little bit along, now, to make it easier, I've split it up again using spaces as the reading frame. Now, have we improved that information?
Well, it becomes meaningless in English.
Right. Yeah. Yes, that's right. We've garbled it. And so what we've got here now is another extra layer of information. Not only is the code with all of the letters neatly written out, we've got another layer of information. In other words, the cell has to know that machinery, that ribosome machine, has to know where to apply the reading frame. In other words, where to start reading the sequence and in what order. So the meaning is in how the code sequence is read. So again, we've got a property of a mind that's outside of the substance that's being used to convey the information. Now that adds an extra layer of complication. So you saw with Glenn's slide there of how many different combinations you can have of just 4 letters. Well, just imagine how much more complicated it is when you have to read them in groups of 3, but you have to get the right groups of 3. And because they're all stuck together in one long sequence, you have to start in the right place. Otherwise, the whole thing is rendered useless. So let's go back to RNA and protein. So here was our problem. We have 4 bases, but 20 amino acids. Our solution, 3 bases, will code for one amino acid. So we have to apply a reading frame, and that will tell us what amino acids that need to be strung together. So those words at the bottom there are actually just a shorthand for the names of amino acids if we apply the reading frame as it is there. Now, imagine what would happen if the reading frame was just shifted. You'd end up with different amino acids, which means you might end up with a different protein. And most of the time, you would end up with a useless protein because the structure and the function of a protein depends on getting the right sequence of amino acids in the right order. So randomly changing DNA letters is not going to help. In fact, it's going to muddle things, just like my little sentence about the cat running off the mat. Right. Now, when cells are constantly dividing, the information has to be copied accurately, and the same with the reading frame has to be applied accurately. Now, when you're copying information, you think about that. It's quite easy to get typographical errors. Now, typographical errors do not add useful information. In fact, they degrade it. So the wrong amino acids, if we're reading our sequence wrongly, or some of those letters have been transposed or left out or knocked out, you end up usually with a useless protein. So it is very important that information is accurately copied each time a cell divides. And if you look at the process that's involved in that, it is hideously complicated. Here is a simplified version, and I can assure you this is a simplified version. The number of molecules that's involved in copying information, making those 2 complementary strands come apart and get copied into 2 accurate copies is very complicated. And if we just have a closer look at the first part of that, during that process, the information is actually copied, but then it is also checked and edited. So it's as if you've got a proofreader over your shoulder watching what you're doing if you are copying information. Now, that is really brilliant. So we have a built-in system for copying, proofreading, editing any mistakes that are found straight away during the copying process. Now, even after the information has been copied accurately, it can get damaged by various cellular processes that happen, or various things that come in from the outside. And so your DNA is being constantly surveyed to make sure that it is remaining intact. And there are built-in repair processes. So there's constant surveillance of your DNA. This is going on all the time in your millions and millions and billions of cells in your body. And any mismatched or damaged DNA can be repaired. And again, you have to have, this is a very, very simple explanation of it here with these diagrams. There are dozens of molecules, and they're huge, complicated molecules as well, involved in this surveillance. And if a damaged piece of DNA is found, molecules will come along, the repair gang, as it were, will come along, take out the damaged or mismatched bits, and put in new bits using that base pairing as the template. Now, sometimes the DNA becomes just hopelessly damaged and it can't be repaired, which means the cell effectively becomes non-functional if the DNA is too badly damaged. And there is actually a nuclear option, as it were. If that cell's DNA is too badly damaged, the whole cell will be dismantled. And that is a process that's called apoptosis, which just literally means falling over. But if the DNA is irreparable, a whole cell is dismantled and will be replaced from a clean copy, as it were, from elsewhere. And what happens is that the cell literally starts splitting itself up, and it will shrink and shrivel up and become fragmented. And so that way our cells, the integrity of our cells is maintained most of the time. Now, some diseases, particularly some cancers and some other diseases, result of this repair process being knocked out. In other words, the surveillance and repair mechanisms get knocked out, and that's where we get a lot of diseases from. But healthy tissue is being constantly surveyed and repaired. So let's summarize what we see in the way genes work. Right, we have layers of information. It's not just lots of information. We've actually got layers of information in terms of how the DNA and RNA letters are put together in sequence and how they are read, which means that has to be built into the cellular machinery that reads it. Information is a product of a mind. It's not the product of matter and energy. All those C's, T's, G's, etc., they didn't make that information. It's the way they are organized, and they can't organize themselves. Once they are put in the right place, they will stay there because of the chemical properties, but they won't put themselves in the right place. Now, when the information is being copied, there is a copy, check, edit system that will maintain the accuracy of the DNA in cells and maintain the accuracy of the RNA. And there is ongoing surveillance and repair for most of the time. It's loss of that surveillance and repair that results in genetic diseases. That's not evolution, that is devolution if you lose that repair process. So healthy cells are designed not to evolve. DNA is not designed for evolution. In fact, it is designed to stay the same. Now, as Glenn mentioned, in the Bible, we are told 10 times that God made things to reproduce after their kind. And that is built into DNA. DNA is designed for after their kind. It's masses of information set into a whole lot of complicated cellular machinery which keeps it the same. It's designed for after their kind. And I think we've got some nice examples of that to look at. I think Craig's got one as well. So what do we see out in the real world? Well, we don't go around with a microscope looking at DNA, but we do see the results of DNA being designed for after their kind, for things to multiply after their kind. So if we can come back to us now. Do we have any questions or other things to thank yous before we go on?
We do indeed, yes. We'll take a few questions now and some thank yous before we carry on. And we've got a little example from Craig in just a little bit. We do have some questions. First off, very, very briefly and easily, Diane, you can answer this one. What is Diane's scientific background?
Oh, all right. My background was originally in medicine. I did work in clinical medicine for a few years, but then for various circumstances, I went into medical education, and I ended up as a lecturer in biomedical sciences at the University of Canberra. But I'd worked in a couple of other universities as well before then, and eventually into creation research. So.
Yeah. Nice and easy. Very good. All right, a few quick thank yous then. Thank you very much to Doki Doki for your donation, and to Douglas as well. Both of these came in before we'd even started the program, so thank you very much for that. That's much appreciated. As well as to Iron Matt as well for his donation. And yes, we do really appreciate your support. There's another one from Doki Doki as well. We do very much appreciate your support as we go through these programs. So thank you very much. Well, another question that came through actually before we even started the program. So perhaps we want to ask this and maybe spend a little bit of time on it, because we've got two questions here from Douglas Boffey, sort of in sort of similarity to each other. But we'll start with this one. Doesn't the 98 to 99% similarity between human and chimp DNA prove evolution? Diane, perhaps you'd like to kick that one off, and then we'll go around to some comments from the others.
Well, 98 to 99% of what? This is where understanding that DNA is information is important. Very small changes in the letters of words can result in huge changes in the actual meaning. So simply because you have a very similar number of A's, T's, D's, and C's, even in similar sequences, does not mean that you are producing exactly the same results. Just like very small changes in words can result in quite spectacular differences in meaning. So, and that's where typographical errors can be extremely funny sometimes. But that's no longer being used as a comment these days, because people are starting to understand the importance of information. The original 98.4, I think, comes from a book that was written before the human genome and the chimp genome were actually sequenced, and it was worked out from just getting strands of human DNA and chimp DNA and seeing how well they fitted together. And sometimes they do and sometimes they don't. But if we go back to our analogy of building things using information, if you were to write out all the instructions for building two buildings, sort of, a brick shed and a castle, well, a lot of the components will be the same, right? They might be both made out of bricks. They might both have windows in them and things like that. So if you were to write out all the information on how do you build a window or a door, or how do you make bricks, that information will be the same, but the results are very different because of the control. In other words, where do you put the windows? or where do you put the doors or the roof and things like that. Now, the amount of information that's needed to say, put this window here or put it 5 feet away is, if you wrote that out in words, it would be very similar words, but the results would be very different. So simply because we have a lot of the same information just shows that our cells are made out of a lot of the same stuff. Well, in fact, if you looked at the information there is in, say, something like a plant, there's a lot of information there, which is the same as the information in a human body, because there are certain things that all living cells need to have. But that doesn't mean you're 50% of a plant. And so you've got to think, that's why I said, 98% of what are you actually describing? And when you get back down to looking at how information works, it's not quite as simple as, well, yes, we've got all these letters that are same, and we've got 98% of these letters that are the same. How are they organized? And what are the letters actually telling you to do?
Well, correct me if I'm wrong. Correct me if I'm wrong, Diane. But doesn't that statistic exclude junk DNA? And it's now only at like 70% or something like that. And the junk DNA now is not recognised as junk DNA.
As
it
junk,
excludes
that's
more
right.
than that. They only compared the 2.4 billion letters that actually matched. They went through and selected only the sections that matched. And so they only compared the DNA that matched. They excluded 1.3 billion letters that they knew did not match. So the question really is, why didn't they get 100% match? They didn't get 100% because they weren't perfect in their selecting of what strands they were going to compare. So it was a total fraud, and that's why they don't use it anymore. But it's still being promoted.
Yeah. So
yes, there is an interesting study that was done quite a few years ago. Genes are actually packaged into what are called chromosomes, which are just long strings of genes. And so someone thought, well, it'd be a good idea if we compared the chromosomes, one chromosome in a human cell and one chromosome in a chimp cell. And they did it with 2 very small ones, lined them all up. But then as I did in my little slideshow there, you've not only got to look at the information, but you've got to look at the outcome. What is that information coding for? And very small changes will make quite big differences in the amino acid sequence that is being coded for. And so they did a study where they looked at a very small chromosome, human example, chimp example, sequenced it, and worked out what results would you get, what proteins would you get out of this chromosome. And there was an enormous difference. It was about 82% different in terms of the outcome. And that's what matters. Now, we did write that up in our newsletter. It was quite a few years ago. So if you go to our fact file and just look up chimp genes, you'll find that report. I can't remember the exact reference off the top of my head, but go to our fact file. It is described there.
And that's why it is very similar to computer programming, because I can tell you, if you got one little dot off in my codes in those punch cards, it's totally different and it's totally wrong.
Yeah, well, you can use statistics in any way, can't you? Like 90% of children like to climb trees, so they must be little monkeys.
I think you've hit on the nail on the head there. Statistics is it's not the father of lies, that's the devil, but it's certainly the offspring of the father of lies. And this is the point that John made. If you've seen the interview that John had with the BBC, the one that got him banned from the BBC, and it's an amazing interview. It's up on our YouTube channel. Go and check it out. But the interview actually brought up this very point, right? Surely, the fact that we're 98% similar in DNA to a chimpanzee shows that we must be related to them. Well, aside from the point, as we've just discussed, that 98% statistic is effectively false, provably false, because they ignored a huge amount of the DNA in it. Let's just assume for the second that we are 98% similar, right? Now, you know that there is some difference between us and a chimpanzee because there isn't a chimpanzee sitting on our panel tonight, right? The fact that we are able to converse, the fact that we've built up a civilization, the fact that we're able to write books and cook dinner and all this kind of stuff shows that there must be a significant difference in reality between us and chimpanzees. Now, that actually, even if we are 98% similar, shows an enormous amount of design that has gone into the genome to be able to make such completely different creatures that are 98% similar. So even if we are 90% similar, we're still evidence of amazing design. And you can go one step further and link it into the way that we've been working with codes, right? Glenn's spoken about codes. Glenn has spoken about codes. Craig has spoken about building castles. Diane's spoken about the design of the DNA and the like, right? And the example which John used is very good. You can take a statement, much like we had a statement with Diane earlier, right? With the cat, and you muddle it up. Well, you can go one step further because you can get the statement, God is now here. You can make one deletion of a space. You can have 100% of letters in 100% the same order. You delete one space and you get God is nowhere. And that's a 100% opposite meaning of God is now here. And it hasn't even changed any of the order. You've just deleted one space. It's a tiny fraction of a change, and you can create an entirely different meaning to your code. And then really, if we are 98% similar, the final question that you should ask is, compared to what? Because just one deletion, if it's only a sentence long, 98% is quite significant. But we now know that there are, 3.1, 3.2 billion base pairs in our genetic code, right? That's an enormous amount of difference in just 2% all of a sudden. And so this whole 98% similar argument, it really just doesn't hold any water, even if we are 98% similar, which we're not, and we've just discussed that. So there's a whole load of stuff that we could delve into here. But Diane, just remind us, where can people go if they want to find out more about the fact file and these kind of reports?
You can go to our main website and just go to what's called the fact file, or you can go to it directly, creationfactfile.com.
All right, great stuff. Thank you.
If you want to get them before they get onto the fact file, you get our newsletter and you'll get them. You get them straight away. So the fact file is an archive of our newsletter. Now, when you look up the fact file, you don't need to know when they were originally published in our newsletter. You can just do a normal keyword search and find them.
That's great stuff. All right. Here's another question then from Sandy C, and we'll ask this one. Do you think the origin of life researchers are getting close to creating life in their labs?
Well, there's an old joke about the scientist with his bottle of brew of primeval soup with some sort of creature emerging from it. So, well, if I can create life out of this test tube, I will have proved that it doesn't require any intelligence. So even in that question, there are some underlining assumptions. Now, I'm not scoffing at you, Sandy. It's just that is, as with those quotes that I used in my slides, deep down, scientists do recognize that there is information there. They just don't want to take the next step and think, well, where did the information come from? It didn't come out of the molecules. Information comes out of a mind, and they don't want to put those two things together.
Well, if we're ever asked what would convince you that creation is not true, what makes creation falsifiable, I guess, then my response would be, well, if you could show me that DNA can generate from natural processes, then I'd have to start questioning whether God had put it together. But I'm very confident that that's never going to happen.
Yeah, because it's been attempted since the 1950s with, as I think Diane was alluding to, very well thought out, very engineered, a lot of intellectual design experiments, and they've all failed. So,
And as it's interesting, right? When you look at the last time that human beings were all together as one with one language, which was of course the Tower of Babel. God came down and saw mankind, and He said, "Look, the people have one speech and one accord. Now, nothing that they desire will be withheld from them." And so God brought down judgment, split up the tribes and nations, and off you go. But it is sort of interesting that who knows, maybe one day, as we all come together as one accord and we all speak the same language and we're heading towards the so-called, end times, without getting into eschatology, maybe we will find out a way to be able to create something that could be regarded as life. But even if we do, that's still not going to prove evolution, because it requires an intelligent designer to be able to do that. Now, I'm not saying that will happen. I'm very skeptical of that. But even so, it would prove that it would take an intelligent designer. It would be us attempting effectively to be God. And all we've just proven is that it required a God to do it in the first place, right? So it has to be further than just showing that a scientist can create, life in a lab. It has to be evidence that it could happen by itself, with no outside intelligence required, no special circumstances or anything like that. Otherwise, you're just proving that a God had to be there in the beginning. So you're back to the point of a creator.
Yes, I think Craig cheated, because he didn't start with just basic chemicals. He started with Lego blocks.
Yeah.
You need to go back to a big jar of oil and just shake it up and down and hope that
Yeah.
And then we'll ask, where did you get the oil from?
No, I'll get sore arms doing that.
Good stuff. Right, let's do one more question before we go over to Craig's a little bit to sort of finish us off, and then we'll finish up with a few questions. There were quite a lot of questions that have come through, and a couple of quick thank yous then beforehand. I'm just checking to see if any others have come through, which they haven't. So we've just got a couple more thank yous very quickly. We've got a thank you from Doug Murphy. So thank you very much for your donation. And I thought I saw another one. I did. It's Doki Doki again. So thank you very much there as well. That's great. Okay, let's have another question here then. One that's a bit more related to Diane's program a little bit earlier, which is this one. Does the cell get new information from each frame shift? And is it true that the cell reads the DNA backwards?
Well, yes, DNA can be read backwards. That's the whole point of the of using the framework, as it were, to read it in threes. There are indications in the DNA strand as to where you start and which way it gets read, because you've got two strands, so you could store information on each one separately. And use that, because it won't read the same. It's not symmetrical. It won't read the same if you read it backwards and forwards. So again, that just gives you an indication of the brilliance of the designer to put meaningful information in both of those strands. After all, how many English words can be read backwards and be meaningful? You'd have to be really clever to be able to do that. Yeah, there are some. They're called palindromes, and they're quite interesting. But just imagine how clever the creator is, how clever God is, to be able to actually embed that sort of useful information both backwards and forward. And that means that you can store more information within the same strand, within the same amount of substance, as it were. You can really pack it in. Now, where does it get new information? Well, effectively, you do have new information, but that doesn't mean it's useful information with each frame shift. You do have new information, but it's not necessarily meaningful information. You've got to look at the end result of that information. Is it something that can be put to use to build a useful protein? And quite often it's not. In fact, most of the time it's not.
Yeah. And I like what Sandy just put up on the chat. Origin of life research is just a complete waste of time and money.
Yeah. And you do have to question the ethics and the morality behind it, in the sense of, playing God ultimately. It's similar with some of the genetic modification and so on and so forth, and the cloning and the like. It's like, how far do we have a right to play around with this stuff as humans? And half the time, it's not even there to try and help people. It's not even there to try and advance medicine. It's up there with the sort of, search for extraterrestrial life. It is purely pointless, right? And it's purely to try and find a savior other than Jesus Christ. So it is. You could almost
say spend billions doing that, don't they?
Absolutely, yeah. So it's it's something then to think about. All right. Well, we've got about sort of 20 minutes left or so. So, Craig, why don't you finish off our last section for this evening, and then we'll come back and do a few more questions before we round things up. So, over to you.
Okay. Well, if you'd like to put my slides up.
Yep, we're up.
Okay, so I've just a very brief sort of look at the coelacanth. I've just been spending a little bit of time this week looking at them. So the coelacanth in 1859, when Charles Darwin wrote his book, was believed to be 400 million years old and an extinct creature. It's a lobed finned fish, so it's got the sort of extensions on its sort of fleshy extensions on its fins. And they believe that these extended fins mutated, the genes mutated over time, and they led to the walking fish, which then, of course, led to things like reptiles, birds, and mammals. So us. And the scientific name for the coelacanth in the fossil record is Coelacanthus granulatus. So that's 1859. At the time of Charles Darwin. But in 1938, the coelacanth was found alive in South Africa. And this is a picture from Wikimedia Commons that shows that a fish in its habitat. But this living one, since it's been found, they've now found fossils up to as recently as 66 million years ago, which is still an incredible amount of time ago. And it's found alive, of course. It's still lobe-finned. It still has those sort of extended fins. And they still believe that gene mutations lead to walking fish. But these creatures live at 160 metres of depth, which is about 500 foot for our American friends. And no one sort of believes that this fish needs its fins to walk in the habitat where it's now found. But they believe, from speculation on the fossil record, that it once also lived in much shallower seas and estuaries and so on. But it's argued as evolving 6 times slower than other creatures. And because they think it's still evolving, they've given it another completely different genus and scientific name, Latimeria, after the scientist by the name of Latimer that found it, Latimeria chalumnae. So that's the modern one. And it was very interesting to read this comment by a scientist and in an article that lobe-finned fish, like the coelacanth and lungfish, are known to zoologists as sarcopterygians, meaning fleshy fins. Tetrapods, including people, are descended from this group, and the coelacanth is more closely related to people than to other fish, which I found incredible that they can't let go, that after 400 million years, a coelacanth looks like a coelacanth, yet it's more related to us than other fish. And this Dr. Alex Meyer, who I'm sure is a very brilliant man in his own field, but comes to the wrong conclusions, that evolutionary speaking, we, us humans, are actually sarcopterygian fish. Now, this is the sort of nonsense that you can come up with. That 400 million years, I mean, just try and comprehend how long that is and how evolution can't be explaining anything of coelacanths evolving into walking anything. And this is a model that we're getting made of the coelacanth. It's not finished yet. It needs to be painted and so on. and I'm excited to get that for our museum. But the coelacanth proves, the evidence out there has proved once a coelacanth, always a coelacanth, and that's what designer genes do. The designer made genes for a coelacanth, and coelacanths have always done coelacanths. So it has designer genes. And just the fact of changing the genus name, as the evolutionists want to do, doesn't mean that evolution is actually happening, or that the genes have unlimited capacity to change. And ultimately, to humans, we're regarded basically as coelacanths or a lungfish. And finally, we recognise that model has been made by somebody, and basically it's only fiberglass and a bit of paint ultimately, and we immediately recognise that as being designed by somebody, because fiberglass and paint don't do coelacanth models on their own, and proteins, amino acids, even if you got them, they don't do living organisms on their own either. So that's just a brief example of a living fossil, and where the scientific world is at. They're still saying that they're evolving. They still believe that they evolved into walking fish and then walking creatures, even though the evidence for 400 million years in their time frame is that coelacanths don't change at all. Okay, come back to us, please, Joe.
Absolutely. And just a reminder as well, by the way, folks, let's bring us back together. This whole point of living fossils, you do need to take it in all of its context, because if you look at just purely the names, then, well, it makes the appearance that these creatures have evolved. It makes the appearance that these fish have changed. So if you consider what is now known as cockroalites, which is one of those fish from the Green River Formation in Wyoming, they are identical in every way morphologically to the freshwater sunfish, right, that live in the United States and so on and so forth. And yet they've recently just undergone a name change to give the appearance that they are a completely different type of fish, a completely separate type of fish. You see this as well with things even like the ginkgos, right? Even though they're still called ginkgos, the Jurassic ones were known as Ginkgo huttonii, named after James Hutton, right? And the modern ones are Ginkgo biloba, even though we've been able to actually isolate the gene that has caused the minor morphological difference between the two things. And so really they are the same thing. They are the same kind. And that's the really important thing to remember, is that God didn't create things according to their species. Species is something that mankind has implemented onto the classification system in order to help it make sense to us. It was done by Carl Linnaeus, and it was actually based off of a biblical principle, the idea that God actually did create things according to their kinds. So in the Old Latin Bible, the word kind was genus. So Carl von Linné, who invented our sort of modern classification system, particularly the binomial system, the two naming system, he said, God has created things according to their genus, according to their kind. So I'm going to create that. But it's clear that there's lots of diversity within a kind. So we can say that there is a special type of that kind, which is where we get our concept of species from. So beware of this idea of species. Remember to go back to the biblical concept of kind, which is built into our classification system. And also remember, what was the name of Charles Darwin's most famous book? On the Origin of Species. Now, if he'd have made On the Origin of Kinds, I'd have been a lot more impressed, but he didn't. So just bear in mind that when you're looking at evidence of change within a kind, it doesn't necessarily equate to evolution, which is different kinds evolving from other kinds. So you need to keep that all in mind and bear that all in mind as we go on forward.
The megalodon's another classic example, Joe.
Exactly. Yes.
That they refer to as an extinct great white shark.
Well,
not just
and they've changed that differently as well. See, this is some politics which is going on at the moment, and I need to write it up right. So the megalodon used to be referred to as Carcharodon megalodon. Okay, now if you know your Latin names, you'll know that Carcharodon is the same genus as the great white shark. right? So if you go to the Houston Museum of Natural Science, which I did last year, they still show you the huge, great big megalodon, Carcharodon megalodon, and they show it next to a tiny little great white shark in comparison, and they say, look how much sharks have evolved. They used to be big, now they're little. Right? No, I'm afraid you're using the wrong term there. A shark has devolved for sure. Now, sharks are interesting because they're cartilaginous, right? They haven't got any hard, bony structure to them. Their hardest parts are their teeth. And it's their teeth that can actually show you, relationships between shark species. And so if you compare a great white shark tooth next to a megalodon shark tooth, they're pretty much identical, except for, of course, the size. One is a lot bigger, one is a lot smaller. But you'll find that there's been a group of scientists recently pushing for megalodon to be classified as a different type of creature, a different type of shark. And you'll find the reason to this is that they want to push it towards a genus known as Atotus. Now, I know a Atotus shark. I've dug them up from places, they're found all over the world, but particularly found in places like Morocco, and we have brilliant specimens of them. But they're much, much smaller than the megalodon, and they're supposed to be some 70 million years older than the megalodon. You see, they came to realize that it doesn't make sense evolutionary that sharks used to be big, now they're small. So they said, well, we need to make this as a different connection. We need to find a small shark that's supposed to be millions of years old and claim that the shark evolved from a small tooth shark into a big tooth shark, because that's what evolution shows. Even though Atotus shark teeth and megalodon shark teeth look nothing alike, they're now being argued as being the descendants of this ancient shark type. Well, I'm sorry, but I know that John agrees with me and will continue to make the claim at creation research that the megalodon is just a very large great white shark. Carcharodon megalodon, I think you got the right classification in the first place. They used to be big, now they're little. They haven't evolved, they've devolved, they've gone downhill, but they have reproduced after their kind, just like God told them to do in Genesis chapter 1. So keep looking at that, because we'll need to probably write that up at some point. All right, let's see if we've got any other questions. I know we've got a lot of different questions and stuff come through, so let me just go to our pinned chat. And we have a thank you as well from Doki Doki. I don't think we've done this one yet, so thank you very much there, Doki Doki. That is fabulous. We've done that question, so let's go to this question here. And I think this is perhaps more of a reference to Richard Dawkins' comment about design, bearing in mind that Richard Dawkins just in the past week has claimed to be a cultural Christian, which is all very interesting. But anyway, let's see how we deal with this. Is it just a coincidence that all the biological world looks designed? I think Dawkins famously said that it gives the appearance of design when it isn't actually designed at all, of course, in his opinion. So any comments on that from the team?
Well, it's a bit like the duck theory, isn't it? If it looks like a duck and walks like a duck and quacks like a duck, well, it's not a pterodactyl. It's a duck. So, yes, the biological world looks designed, and the evolutionists sort of occasionally give themselves away when they use the language of design, because it looks designed because it is.
Yeah.
And anyone can recognize something that's designed. We all know the difference between things that are designed and the results of chance random processes. And we spend our lives clearing up the results of chance random processes and trying to reestablish design. So, yes, the biological world looks designed because it is.
But I think Greg made the point, it can look like a fish, but it's still a human, right? It looks like a coelacanth, but it must be a human, We must look like a fish to them. Sometimes there's no amount of reasoning. We keep making the point over and over again. Design takes a designer. And when you look at the DNA, it is very clear that can be no other way. It's so complicated, our mind can't comprehend it. Diane, in your talk, the thing that I was most amazed with was, it's even designed to go in and look for errors and correct errors. Just as a computer programmer, I mean, that takes a lot of intelligent design and skill. That's what I spent a lot of time doing. You get something that doesn't work out, and it doesn't produce a result or produces wrong results, and you've got to go back and figure out where was it off in the computer program. That takes a designer to build a system that does that automatically, and to know that it would need to do that.
When you look into it, there's no such thing as apparent design, because design always implies intelligence, intent, and purpose in the product. So, for example, if you're looking at a pattern in the sand, well, that's only a pattern that's come about by the natural processes of water and wind and sand. But when you look at true design, real design, it's always something like that castle example, where it's plastic won't do building blocks on its own, and building blocks won't do castles on their own. It always implies outside purpose, intelligence, and intent to get a product that has some meaning.
And time is the worst enemy, because the second law of thermodynamics, things break down, they don't improve.
Well, that's exactly it with this example with the building blocks. And I ask 5-year-olds, I say, if I did that for a week and shook that, would it start to come together as a castle? And even a 5-year-old intuitively knows, no, that won't happen. And if I did that for 1,000,000 years, what would happen? Well, it'd be a big pile of dust at the very best. It would never come together as a castle. Time doesn't help it at all.
And that's what we found as well, wasn't it, Glenn, when we were doing programs in the States, and we had a sort of a dinosaur Lego model with exactly the same point, right? That you have been making, Craig, with the castle. I know you have a model of the Sydney Opera House in the museum, but you just see people's eyes kind of light up, and they make the connection that, oh yeah, even something as simple as a Lego model couldn't put itself together, and that's ridiculously simple when compared to the complexity of DNA, and that in itself is just one tiny component of the complexity of a cell, which comes together to create the complexity of a body, and it just becomes mind-blowing very, very quickly. So Diane, I don't know if you can answer this question. This was a question that Sandy asked about an earlier point that you had made, the name of the article on the Creation Research fact file that you mentioned a little bit earlier about the DNA. I think it was about the DNA percentages, 98% and the like.
Oh, the one about the chimp genes.
Yeah.
I think if you just look up chimp genes, or I'd have to go and find it. But if you go to the fact file and just put chimp genes in the search box,
that should come up. That's fine.
It should come up.
That's great. Yeah.
I think that the result they got was that if you looked at the outcome in terms of what the genes are actually coding for and what you end up with, it's a sort of 82% different. It's a huge amount of difference, and that was just one small chromosome. I don't think they bothered doing it for all of the other chromosomes because they were so put out by the results that they got.
That's good. Let me ask this question then, and then we can perhaps begin to wind things up. And so I'll start off by giving a comment and then throw it over to the others. Should modern corn be credited to God, evolution, or man? And you could really ask this question about so much modern agricultural plants, right? Things that we consume for food, not just corn, but also wheat. If you compare what the wheat was like back in the Middle Ages compared to what we have today, it has been engineered, shall we say, by man to effectively be suitable for our machinery and to produce a bigger crop. You look at the fact that carrots are orange now. That was engineered purely around politics. So it's some interesting history to some of this agriculture, but it should be pointed out that agriculture is basically the biggest experiment experiment that human beings have ever done, some 6,000 years worth of known history, bearing in mind that the first thing that God told Adam to do was to tend the garden. That was the first job that he was given. So agriculture has really been the longest experiment that we've been doing. And the amazing thing is that we've been able to make corn fat, we've been able to make wheat short, we've been able to turn carrots orange, but they're still corn, they're still wheat, and they're still carrots, right? So the first thing is that the ultimate origin of this wheat, the ultimate origin of the carrots and the corn has to come from God, right? In the beginning, God created and he said, let the earth bring forth plants. That's the ultimate origin of these plants. And he told these plants to reproduce after their own kind. And that's what we've seen. And we actually have a program, I believe it's also on our fact file, Diane, about cabbages, because Richard Dawkins actually mentioned cabbages. And we found examples of wild, I mean, you compare cabbages, right? The cabbage that you eat, the broccoli that you eat, the cauliflower that you eat, the Brussels sprouts that you eat, the kale that you eat, the kohlrabi that you eat, it's all the same species. It's Brassicarola sera. Huge amount of diversity in just one single plant. But if you leave it to its natural, state and let it, sprout and produce seed and reproduce, it'll eventually revert back to its normal state of a wild cabbage. So yes, it does take some selection to actually keep it as a cauliflower, much like it takes some selection to keep corn as our modern fat corn and wheat as our low sort of growing machine bred wheat. So there is an element then of man coming into it and selecting certain aspects, selecting certain properties, hence turning the purple cabbages and purple carrots and white carrots into orange carrots. That's a case of us selecting things and only sowing certain seeds and so on and so forth. So you could say that the particular breed, the natural selection, if you like, becomes artificial selection. We are artificially selecting these things. Then you get to the other interesting thing when it comes to things like genetic modification, and that's particularly in corn in the United States, where we've taken a fish gene and put it into a corn in order to stop a certain parasite, a type of worm, right? Now, without getting into the discussion of the ethics or morality of that, you do realize that all you've proven, if you want to create new genetic material, is that it requires an intelligent source. And that intelligent source didn't even make the genetic material in the first place. It just took it from somewhere else and put it in, right? So all you're coming back to is this very important point that you have to have information. Genetic material has to come from an intelligent source, and it requires an intelligent source even to just modify it so-called unnaturally, right? If you want the corn to have a fish gene, it will never evolve its own fish genes. You have to do it for it, right? That's without getting into the ethics of it. And so, yes, number one, you can credit the creation to God, the instigation, if you like, to God, the created after its kind. Some of the diversity within a kind is down to us selecting for or against, but there's still no evidence for evolution in it, even if you end up with a corn with a fish gene. Any other comments, folks?
Yeah, I wouldn't mind. You go, Glenn, if you wanted to say something.
No, I was just shaking my head in agreement.
Well, I just think something from the scripture really answers this question to the point as to who should be credited with this. If I can just read from Revelation 4 and just 3 verses. But whenever the living creatures give glory, honor, and thanks to him who sits on the throne and who lives forever and ever, the 24 elders fall down before him who sits on the throne and worship him who lives forever and ever. They lay their crowns before the throne and say, "You are worthy, our Lord and God, to receive glory and honor and power." Why? For you created all things, and by your will they were created and have their being. So it's pretty clear who should be given the credit.
And you find a funny little dilemma as well in the case of Professor Andy McIntosh. If you remember, he came on our show a few months back, Ryan, talked about bombardier beetles. And his research on bombardier beetles ended up going on to produce a patented design for a sort of an injection, model, right, which was based off of the Bombardier Beetle. And of course, when his, the patent office and his supervisors and the guys in charge at the university said, well, who are we going to credit with this? Because we can't really credit the Bombardier Beetle. They weren't particularly keen on Andy's suggestion of crediting the creator God. So it does, and this is, he genuinely wanted it to be credited to the creator God who actually invented it in the first place, because as Andy said, all we've been doing is copying it. So thumbs up to Professor Andy McIntosh for doing that. But it does, yes, I completely agree with you, Craig. Credit needs to be given to the creator, who is Jesus Christ, who we will all fall down on our faces in front of when we come to face with him. Well, it's about time that we start wrapping things up. I know there's one or two questions that have come through which we haven't got a chance to get to, but I think we've pretty much covered all of them. So thank you very much, everybody, for your interaction. That was fantastic. And it's been a pretty good program, despite being short of both John and Sam, but that's all right. We should be back next week. We will be back next week, and everybody should be back next week is what I mean. So we look forward to being back together then. Can
I answer Doki's question?
Of course you can. This last one that's just come through. No, there you go.
No, you can't.
Can I cut and paste eagle's wings DNA into my own so that I may fly? I think that you might end up badly disabled if you attempt that, but there we are. Thanks for that, guys.
And we don't get wings when we go to heaven.
We're not going to be sitting up there on a cloud.
We get harps.
Harps, yeah.
Yes. But it would be nice sometimes to be able to fly.
Yeah. All right. Well, folks, it's time to bring that to the close. We'll have another great program for you next week. Be sure to join us then, where we should be back with the whole team, John included. So thank you very much for all of your interactions tonight. Goodbye and God bless. And we will see you soon. See you later, folks.
Goodbye.